So can the end of the tape touch the bottom edge corner? Can you? You can. So if you can touch this, you can realize the actual dimension on the substrate. Okay? The question is, it takes us maybe a few hours to attach a tube. If you are lucky. If you are lucky, we fail for 1 month. Attach one tube on the probe. Okay? So this is a very expensive way to do it. Nano pencil. We say graphite is what you have inside your pencil. And nanotubes. The shape is like graphite, one sheet of them, but you roll them up to chip structures. And actually you can do it this way. Right? You have a substrate. You coat it with a polymer that is sensitive to electron. Okay? And now you bias your substrate to a nanotube. You apply a voltage. Then the electron will start to emit and strike on the polymer. Right? If you do that, you can scan your nanotube and write any pattern you want on the substrate. Can you see this? Nanotubes, nanopencil, and this is 1 micrometer length. So the width of each line will be a few tens of nanometers. So this is nanopencil. The question is, can you use this technique to write an encyclopedia on a piece of substrate? How long will it take? So unless you can realize with a so-called parallel processing technique, you can write the computer control at, you have 1,000 pieces of probe and write at different speed. But now the probe will fight with each other. You cannot get too close, right? So that's one of programming. Alright, chemical and biological probe. Now, this is a piece of nanotube, right? If you cut the ends here, you have some of the atoms, carbon atom that is not bound to others carbon. So they are free. We call this dangling bond. Okay, this defect. And now what happens is that you attach these free carbon atoms with the types of molecule that you know. Okay? For example, you know about DNA, right? Inside DNA, you have many sequences of the nucleotide, right? You have T, A, G, etc. Okay? And you know that all these will only bond to specific nucleotide, right? Okay, T will bond to A, etc. Now, if you attach the probe here with 4 kinds of nucleotide, and you use this to scan a single-strand DNA, map them, to find out at which position will be A, which position will be T, etc. Then you will be able to map the sequence of the single-strand DNA mapping. Okay? It looks interesting and promising, but this will take forever, right? How many million pieces of nucleotide on a piece of single-strand DNA? So it will take years to do it. But chemists found that they can do it on a simple approach. They can recognize CH3 or COH molecule code on the substrate, right? Nano-electronic devices. We say that carbon nanotubes can be used as a semiconductor materials, right? They can replace the silicon to build the silicon chip or devices that you want. So this is how the first field effect transistor looks like, right? We have one electrode, source, drain, and the bottom one is the gate. Then you can see the switching, right? But the question is that this is very simple layout, and this structure will be micrometer big. Okay? And the first device will spend months to build one. How many years do you need to build one? CPU chips, which has trillion pieces of transistor. Can you do that? And this is the challenge, right? And it is your assignment, right? Okay, am I doing so fast? Too fast? Can you follow? Or too slow for some advanced student? Alright, in the first talk, right, we talk about nanotubes, right? The atoms are everywhere on the surface, right? So current will flow through only near the surface. So now, if you touch the nanotube, or you attach an alien molecule on the nanotube will tell what kind of molecule is actually attached to it. So this is what we call sensing. Okay? So if you do that, for example, you attach with the so-called antibody, right? And this will be able to use, the receptor will be able to use to sense the protein. Okay? So this is one kind of sensor. And this is a so-called based on silicon nanowire. It can be done on silicon nanotube as well. Okay, flat panel display. I mentioned about this before, right? In the first part. Anyone still having this kind of TV at home? Of course, I'm still having one. Of course, you might use the LCD or plasma. Plasma is not durable. LCD, not bright enough. Okay, not too dark. Okay, so this is a good one. Why? You have one electron gun, and this electron is extracted through the fluorescent screen here to strike on the pixel that you want, and it scans very fast to give you the picture. Okay? Now the question is, you have one electron gun which is so tired to scan so fast, okay, and so bulky. So can you now change it to this configuration? You have many pixels on the screen, right? Red, green, blue. What about having one nanotube or a bunch of nanotubes behind the red pixel and the green pixel and the blue pixel? Every pixel has an electron source from nanotubes. And if you do that, you just have to apply electric field one at a time. Once the electric field is high enough, the electron actually will start to emit from nanotubes and accelerate and bound and strike on the red, green, blue pixel. And then we give it a color. Okay? So if this is the case, this will be your TV, your display. In principle, it can be as heavy as 2 pieces of plastic sheet. One plastic sheet you coat with nanotube, many dots. Another one, you put a spacer, put another piece of fluorescent screen before the pixel on top. This will be your TV. Next time you go to picnic, say, wait a second, let me bring my TV. Let's go. This is my TV. Now the question is, this process is called electron field emission, and it requires very high vacuum level to do it. It is 10 to the power minus 7 millibar order. Can you seal 2 pieces of plastic to high vacuum? This is your assignment. Okay, it's very tough. So next time if you buy a flat panel display, they say it's based on electron field emission. Check with me whether this is reliable. Okay, this is some of the work prototypes demonstrated by Samsung more than 10 years ago. But now Samsung has not sold any piece of flat panel display based on electron field emission. Why? Many problems. Okay? X-ray gun. Anyone go for X-ray before? You do that, right? Medical checkup. You go into a room, you see a big machine, like a car, and you have to shield yourself everywhere. And the guy said, stand there, don't move, and I go out. You stay there. And then the patient goes, boom. Then you're done. The X-ray is penetrate through you. You see all the shadows, see your bones and organs, everything. That's X-ray, right? Okay. It's big, huge. Can you ask a soldier who's going to fight, right? Carry a car at the back, which can be an X-ray. In case they have some injury, they can do X-ray to see whether they have bone injury. Can you do that? You cannot. When they jump from parachute, then you'll die because of the weight of the car. Now, can you make your X-ray gun this big? Nanotech. This is demonstrated. Alright? It's actually about this big, you know.